Hello. Make sure each part of this is working. All right. How are you?
I'm pretty stressed.
Why?
I have an orgo test
Oh,
on
but
Thursday.
isn't it take home or whatever?
No, professor, it's at this thing called lockdown where it like locks down your browser and it records you. And if it thinks you're suspicious of cheating, it literally shuts down your test and you can't take it.
Why don't you just use a second device?
I don't know.
And
Because
just put
I
it
don't
right next to the device you're looking at and behind.
because I don't want to get a 0.
There's got to be a way around lockdown browser. I just can't imagine that something that's free is.
It's just hard. And you can't even go back to the questions.
Well, that's just annoying, but
It's just scary.
well, if you want, before we leave, I can look up. I'm sure.
I can okay, help me cheat. No, I don't. The thing is that like I know the stuff except for some stuff, which is really really hard.
Because if it's good, then I'll use it. Is what I'm saying.
What do you mean?
Like if it's a good system, then I will use it. Like I'll
It's not.
because it stresses you out.
It's terrible.
I don't understand how I'm on spring break at Columbus and I have 15 emails from Columbus. Okay, yeah.
No, 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 una jodera. Eso, no, eso demora mucho para darme.
All right. Because that's the first thing I do whenever I try to use a new system is it any good? And I try to beat it myself. Is it beatable? Then the second thing I do is I let loose, my Columbus kids. Because if it's beatable, they'll find a way. Extra credit, but I don't have them handy like I normally do. I'm just giving everyone a few minutes to come in. Let's see, lockdown browser. And let me tell you, we are keeping Amazon afloat. All right. Let's get started. Wow, good number of you came around here today. Well, welcome to whatever this is, whatever number this is. It's the third time we're doing this. And let's just start off nice and easy with a little bit of a COVID update. There is some good little science that's come around from the, in terms of the virus. And, whenever I can get some science coming in to show us, what it is that's happening with this virus, I like to incorporate it. This is a video that they used. They used a CAT scan, which is think of like 1,000 X-rays. That's a CAT scan, and they used it to recreate someone who was in intensive care in terms of what their lungs looked like. You might have seen this before, but maybe you didn't. So the yellow is stuff that's not supposed to be there, and the tubing inside, if you don't know, that's what your lungs actually look like. They have little bronchioles that come into your lungs. When you have bronchitis, usually you have an inflammation of the tubes in there, but pneumonia would then be clumps of fluid, usually on the bottom. This is upper pneumonia, which is much more dangerous and harder to treat. And it's been confirmed by a number of groups that the damage that's being caused by this virus is considered to be permanent, or at least semi-permanent, where it'll take a long time to recover. So there's where you would get bronchitis, but here's where you start to get into a place where you would get pneumonia. And I'm sure there's a better rendering of this video, but this is good enough for me. They found that the two conditions that make you most likely to die is high blood pressure and diabetes. So usually, again, when you get pneumonia, it would be closer to the bottom of the lungs. It's usually one side or the other. It would be like a clump on the bottom. But this, as you can see, is an upper lung pneumonia. And I don't know what it is about them, if it's the nature of the beast, in other words, the type of virus or bacteria that causes you to get pneumonia, the upper part of your lungs tends to be more resistant, or if it's the location or what, but upper lung infections tend to be much more dangerous than lower lung infections. Dual lung infections, are more dangerous. All right, I think this can go on for an hour, but you get the idea. Well, this is life according to John Hopkins, 3.5 million for almost 400,000, or kissing 400,000. 81,000 deaths with 300,000 recoveries. So that's almost 4-to-1 death to recovery. But again, it would be surprising if that's the number, because everywhere it's hard to get tested material, even in the US. Somebody was mentioning to me that they thought that the pandemic was slowing down. Confirmed cases is not slowing down. Daily increases is slowing down, right? Which is nice. But if it goes, if you're talking between 33,000 to what is that? Just around 30,000 per day. That means you're still growing at a pretty sick tick there. There's also the Florida Department of Health. I haven't seen Miami-Dade County all day. But if you ever want to know what is going on and what is not going on, take a look at the data yourself. It's publicly available. It's not that hard to read. You don't need to be a PhD in something. You can see how the daily increase is going down. So that could be a positive look to it. Another thing to look at would be death rates or hospitalization rates. But if you even look at Florida cases, right, you could have seen this piece here and been like, look, it's going down, but no, it's kind of tapered off there. Is that Duval? I think I had the Dixie, no, Dade. So there's 5,000 cases in Miami-Dade County. 5,100 with 14,500 in Florida. That's a lot. And there's 2,300. Is that Florida or is that Dade? Almost 1,000 people, 25 to 34, who have this virus, or 347. But then again, 347 is pretty close to how many people have it, 75 to 84. So it's affecting both. And since it causes permanent lung damage, I feel like these people, if they recover, they don't care about permanent lung damage as much as these people, because permanent lung damage won't last 75 to 84-year-olds as long as permanent lung damage would affect a 18 to 24-year-old or whatever. The rate of doubling is a number I've been seeing pretty commonly from the New York Times. No, that's not what I'm looking for. There we go. Now here, what this is showing you is the Times has been doing a lot of number of cases per 100,000. So controlling for population, it becomes a very important thing when you're trying to figure out, how this virus is spreading, So for example, for every 100,000 people, we have 120 people who have been confirmed positive. In Spain, for every 100,000 people, they have 30 confirmed positives. For Italy, these have 224, et cetera, right? And you can see then, based on the per capita rate, how countries are faring. South Korea, which is one of the first countries that actually ran into this virus, South Korea and us got the virus on almost the same day, and are both of our first confirmed cases. And they have 20 people for every 100,000 with 0.4 deaths. We have 120 people with 3.9 deaths for every 100,000. So now reasons why can vastly expand depending on what it is you're looking at. So one reason might be the response, one reason might be the overall health of people beforehand, et cetera. At this rate, right, at the current declining rate, or whatever you want to call it, we will be doubling the number of cases every 6 days. That is, if everything stays at the current rate that it is now. That's still incredibly fast. To be doubling the number of cases. Same with Canada, Peru is in 5 days. Some of these countries aren't testing as much. Like the UK is not testing as much, but they have every 5 days. 4 days. India's every 4 days. Turkey's every 5 days. Nigeria is every 2 days, and Cameroon with with Guinea every 2.5 days. I mean, so you can see it's still spreading pretty well. Uh, as long as this doubling rate is going as quickly as it's going, China can expect a doubling every 246 days. Just to give you an idea of how slow it can get, that's pretty slow. Now, I think I mentioned this last time, but just in case, some people are like, well, China's lying about their numbers. That may be true. China's lying about, the number of deaths. That might be true too. They're not testing as much. That might also be true. But based on what we know, that's where we're at. Let's see here. It has the short yet sweet 15-minute COVID-19 update here on April 7th. What I'm interested to see is from those of you who should be graduating this year, let's say, right? I'm wondering how Miami-Dade College is going to handle graduation. I mean, because it could be, wait, it's this one. It could be easy as pie. They just change the date, or they're just like, it's outdoors, it'll be fine, or whatever. Or it could be more complicated than that. 31. And I think we started 31, so. So in case you've forgotten, remember there's a take-home, one exam take-home at the end, covering the chapters we have here now that we are separated from the classroom setting. There's going to be the two in-person tests you've took in the beginning, plus the last test, which is take-home at the end. I'll be sure to make sure the class average the test average will be at least an 80. I'm assuming that since it's take home and that I'm not writing it like some kind of monster, that you will have, I'll have a class average of higher than 80. But just in the off chance that it's not, it'll be curved to an 80. And then we'll add the extra credit for homework and attendance, and we'll go from there. So we were learning about mushrooms, right? We talked about how mushrooms are made up of these things called hyphae, and hyphae form these things called mycelium, and they release enzymes, slurp up what they find, and then absorb it in. There was two types of hyphae, septi hyphae and coencetic hyphae, and septi separate and coencetic don't. The idea of a mycelium is to be able to have a larger amount of surface area to be able to absorb more. Some hyphae have the ability to become obruscles, be able to break into, let's say, a nematode on top as a predator, or into the plant on the bottom. Some of them actually work on the outside of a root cell. They're called ectomycorrhizae. And then abruscular mycorrhizal fungi is the ones that break into. Fungi have this magical figure 8 life cycle where they have a sexual reproductive and a non-sexual or an asexual reproductive cycle. And they're different from plants because when this is more asexual fungi because they use non-sex cells. When plants fertilize themselves, they do sexual reproduction with themselves because they use sex cells to reproduce with themselves. And I know these are distinctions no one cares about, but they exist. Fungi can signal each other using pheromones, and that's how they can tell their mating type so that they run into each other. I wouldn't call it male and female. Plus minus seems to be the calling type for these things. They do this thing called plasmogamy, which is when they fuse the cells, then the nuclei fuse. Sometime later, they become a diploid and they do haploid. They do meiosis and become a haploid right away. We talked about different types, like so. We classified some fungi based on whether we can tell. We've determined their sexual reproductive cycle. If we can't determine it, they were put into one group versus another. And so Deuteromycetes was the name for those nons, like the hidden sexual reproductive styles. Everything came from protists. There's actually this game someone turned me on to where you're recreating life on Earth, and I mean, it's interesting because it is accurate, but you spend so little time as a protist and as a bacteria, it's almost like a forgotten stage of it. But yet that was what we did the longest. You know, humans are what we've done the shortest. I don't know, no one cares. So looking at different genomes of fungus, we figured out that they've been around a while, and they were mutualists way back in the way back. These are major groups of fungi: chytrids, zygomycetes, glumeromycetes, asco, basidiomycetes, and it gives you some basic differentiation characteristics of these groups. Like chytrids had zoospores, and they can do anything, although not every chytrid could do anything. The chytrids that are parasites tend to be a species of chytrids that are separate from the other ones. Zygomycetes, these were the ones with the zygospore, and it looks like a golf ball looking thing. And again, the zygomycetes have zygospores in the zygosporangium, whatever. Everything will have zygo to it. Zygosporangium and the zygomycetes. The good thing is, since the naming system was made with other people in mind, it seems, they named them well. Glumeromycetes, these are the groups that actually have the abruscural mycorrhizae that live inside of a plant cell. So these are very specialized fungus. Ascomycetes, again, everything will be named Asco. They live almost anywhere. Ascomycetes are a major player in lichens. Those are the fungus that group up with the algae to be able to live in places that nothing else can live. Their conidia are asexual. They allow for reproduction, right? So they are themselves specialized to be able to do sexual reproduction. But they don't necessarily have to. They can just do asexual reproduction as well. So it allows them to do either from one structure, which is nice. Basidiomycetes, these are my favorite fungi. I once saw a video where Basidiomycetes, a puffball like this, shot out its puffs very naturally. Ever since then, every video is just someone stepping on a puffball. I won't put you through that experience, but puffballs will shoot their spores out, kind of like this does sometimes with rain, but it'll shoot the spores out to reproduce. And these will be basidiospores because they're Basidiomycetes. These are also all the fungus that you have ever imagined in your life. Now, the shiitake mushrooms, the white mushrooms, all the mushrooms you've eaten in your lifetime, most of them are Basidiomycetes. This is a fairy ring, okay? And a fairy ring is aptly named because the way it's made is a fairy, like Tinkerbell, will jump from spot to spot here, sprinkling its fairy dust to allow these things to go. No, that's silly, right? There are no fairies. So this was actually started right here in the middle. It was one fungus in the middle. And then what it did was it started eating what was there. And once it ran out of food, it made these reproductive structures, but over here. and then the spores spread from there. And then it made a little bit of a bigger circle, and the spores spread from there, and it kept doing that. It kept eating what was there, and then when it ran out of food, it would make a big, and then a bigger circle, and then a bigger circle, and it kept doing that over and over. Okay, until the point where this circle became as, if you left this alone, these spores would shoot over here. It would make a ring, another ring, a little bit bigger than that. So then you have to ask the question, well, why would they call this a fairy ring? It's because back then, and we do this now, just because you can't explain it with science doesn't mean it needs to be explained with something else. Science is fine. Fungus play a major role in decomposition. Decomposition is often underappreciated. We would run out of nitrogen, for example, for decomposition. We would also run out of other trace elements in a usable way because they'd be trapped in the bodies of the dead, right? And it would decompose way too slowly. So ants seem to be able to use the digestive power of fungi by raising them in farms. I mean, your guess is as good as mine as to how an ant figured that out. But the ones that, once one figured it out, it got an advantage over the other ones, and the ones that didn't figure it out, well, they're dead. And that's the way that works. Lichens here are, again, that combination of fungus and algae, and they come in pretty impressive structures and sizes and shapes, colors, and but they all play that primary producer role, being the very, very first thing to live in an area and to be able to create life in that area. And the way this thing reproduces is an algal cell, right, wrapped in fungi. They call it ceridia, breaks off, fly or floats, or depends on whether it's airborne at that point or waterborne, probably waterborne. And then it reproduces from there because the fungi and the algae can both reproduce without sexual reproduction. So, and then it'll grow from there. So, in fact, after a fire, you can usually get, since there's still soil, right? There's still dirt. You can still get things to grow, but where there's nothing that's ever grown, the trick is to get lichens to grow there. 30% of fungal species are parasites, and 10 to 50 of the world's harvest are lost, 50% now, are lost due to fungi, like corn smut, which is well described, and then tar spot on maple syrup, and ergot rye. Ergot rye has the active ingredient of LSD, which is a hallucinogenic, and often people who eat ergot rye will die because it's too much of a bad thing. And this rye is not supposed to be this color, it's supposed to be yellow. So if you grew it, you'd be like, ooh. So ergot on rye is caused by Ascomycetes. 40,000 people died in the Middle Ages. You get gangrene, nervous spasm, burning sensations, hallucinations, and temporary insanity. No? Who's in? Friday night? Come on. All right, now. Let's see here. So this frog died because of a fungus. I mean, it wouldn't be that crazy for something like this to wipe out an entire animal species. It wouldn't even be that crazy for something like this to wipe out a lot of a human species. Fungus are notoriously difficult to treat. Fungus are notoriously resistant to anything outside of antifungals, and our bodies have a hard time fighting them. I mean, hence the fact that ringworm and athlete's foot are still common examples that we haven't become immune to. I mean, at least with virus, like when you get the cold, when you get a common cold, the virus is vastly different from the last common cold to the next. But with ringworm, the fungus is very relatively stable, right? So why haven't we figured this out yet? And there are no vaccines for it. There is no vaccination system that we can use for fungus. Some fungus are used to make penicillin, like there's this fungus called penicillium, which is right? And some are used to make alcohol and cheeses, and they're fine. You can eat them and you'll be okay. So here's a little cheat sheet of fungus, and what they do and who they are. And this is a fungus, the cordyceps fungi that actually can kill at a pretty rampant pace. But we're going to leave that alone for now because the videos that I have for that are better if you can hear them. And since we can't hear them, we won't try. So it's 31, 32, 43. And 43 is the immune system, I think. So, and the immune system is going to be apropos. Yeah. So we'll definitely get into the immune system here, okay? Now, some of you might be like, oh, I want to learn about all the systems, and I get that. I don't blame you. But at the same time, understanding how a vaccine would work, I feel like is useful. Understanding how immunity would work, right? Well, what does it mean that if you get it once, can you get it twice? Like all that stuff comes out of immunology. And the one chapter of immunology covers a lot of it. And since it matters, and you know, you have a reason to, that's what we'll do. All right, so animals are consumers, right? They're eukaryotes. And this is talking about many different animals. And in case you're wondering what this is what happens when you're having a bad day. Like this could be a meme where it's like, I'm having a bad day. The reason why you don't see this thing's head is because his head is inside of this tongue. Anyway. So animals are heterotrophs. They have to eat food. Multicellular, they have tissues. Tissues, if you remember, are groups of cells that perform a function. And organs are made of tissues, and organ systems are made of organs, and an organism is made of organ systems. All right, now, this is where it gets really fun. After a sperm fertilizes an egg, something happens called gastrulation. And gastrulation matters. And you're like, no, it doesn't. Yes, it does. So understand what you're seeing is the controlled division of a cell. It's almost like it doubles in a wave. And what you're going to start seeing it do is kind of roll and spin, almost like when you're folding socks or something. It'll kind of like pinch in, as though it's a circle that's pinching inward. See it right here? It's starting to pinch inward, and then it's going to form a tube inside. Now that's the tube that's being formed in there. That tube is called gastrulation. And the opening of that gastrulation, where it folds in, usually becomes either a mouth or an anus. And then whether you become a mouth or an anus matters. So if you become a mouth, you're one thing, and if you become an anus, you're another thing. Now, I'm not saying like you individually, I'm saying like humans as a group. I hope this is not a picture and actually a video. I got a feeling you're going to give me a picture and we're fighting. I'm not waiting for 40 minutes. Okay, wait, here we go. All right, so this is the surface then of the cell. And you can see the hole here. See how it's folding in on itself there? That is gastrulation, okay? And that the determinant of that gastrulation becomes extraordinarily important. And you might be thinking, well, not to me. But yes, even to you, because that will become your internal organs. And there's a point when you were inside of the womb, or, you know, should you or have you had a baby, that all babies, right, would be inside of the womb and then will have this process occur. And this is when the very first bit of internal organs will actually be created and brought along, right? Now, for us at this layer, at this level, knowing that it happens, okay, that bending in is called cleavage. The blastula is the multicellular, it's like a hollow area. And then the blastula doing the gastrulation is how it becomes, how it goes through that process. And that's the natural, normal development of that organism. Okay, and this happens, I mean, within the first 1,000 reproductions, I'm going to say. So if this was happening in you, you wouldn't even know you were pregnant. This would be pretty early on. So here you have your cleavage, right? It's starting to divide, it folds onto itself. And then here's the gastrulation where it actually starts to pinch inward. And then eventually it gets a blastopore, right, which is the opening from gastrulation, and then you end up with some tissue layers, an endoderm, an ectoderm, and then sometimes a mesoderm too, which is a middle layer. Now, when we start to classify animals, this is what we use to classify them. This becomes an important way of classifying them. And we'll get back to this because the way the cleavage is set up and the determination of the blastopore become vital instruments for classification. So larvae is sexually immature and very different looking from the parents. They go through metamorphosis and they grow up. The metamorphosis and development process is controlled by these groups of genes called HOX, H-O-X, or HOX genes. The HOX genes do a lot of body development, body structure, and when they turn on, when they turn off, how quickly they turn on and off, and how long they turn on and off will drastically change the shape of the resulting organism. Humans have been around for a while. I'm not going to ask you dates. All animals, sorry, not humans, animals have been around for a while. Animals came from protists that are called choanoflagellates. Back in the day, 20 years ago, they were called animal-like protists, but that's a little side. So choanoflagellates, and then a sponge, which, I mean, they pointed out here, you see how the sponge has something that's very similar to a choanoflagellate in it. So here's the choanoflagellate in the sponge, and the choanoflagellate is by itself. So you can think of it almost as a community structure of a sponge. So maybe a sponge is something between the protist and the other animals. I know sometimes we look for a missing link. We're like, oh, well, how do we get from monkeys to humans? Where's that missing link? This might very well be the missing link between protists and other animals. Okay. So animals really became much more diverse during the Cambrian explosion, which is the mass extinction that led to many predators to have new opportunities and created a lot more of families, not families like the Rodriguez family, rather families more like kingdom, phylum, class, order families to be created that were predators. Bilateral symmetry became much more popular with a one-way digestive tract and a complete digestive tract. We had the hallucinogena, which is always fun. Another artist rendition of stuff that occurred back then, which should be scary to everybody here. And all of these have to occur with Hox genes. So like this tank looking thing and the weird worm with the plug in it and whatever the hell that is and whatever this is, all of these things have to be developed through Hox genes. And Hox genes are slow to change because if you change too much, you end up like creating a gap in your Hox gene, and then the Hox gene itself will not hold. So in other words, you'll create an organism that is not survivable. Anyway, so these organisms that came around from the Cambrian explosion had to evolve from those Hox genes changing. And so we're going to look at some ways to categorize organisms, and we're going to look at the body plan, one of which is radial symmetry and bilateral symmetry. We talked about bilateral symmetry a second ago being a big part of the Cambrian explosion. Radial symmetry means it's, like a wheel. It has a center point, and then symmetry can be created through multiple planes. Bilateral is one plane, a left and a right, if you will. So dorsal, with bilateral symmetry, dorsal is the top, ventral is the bottom, anterior is front, posterior is back. So for humans, we have bilateral symmetry, and we consider ourselves, when you're considering our symmetry, we're we're standing straight up with our palms out, I believe, like palms forward. So our dorsal end is our head, and our ventral end is like our legs. Interior is the front half, and the back half, etc. A lot of organisms, a lot of animals have sensory equipment in the anterior end, because most animals are not considered standing straight up. Most animals, since they're not bipedal, they're on all fours. So their anterior end will be closer to their heads. Radial animals are sessile, which means they kind of drift, or kind of swim, but not really. Whereas bilateral tend to move. Doesn't mean that everything that's bilateral moves, but it's pretty close. Radial animals typically move actively. Who cares? Animal blah, 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 blah. So we have there's tissue layers for animals. We talked about the endoderm and the ectoderm, and we mentioned the mesoderm. So ecto is the outside, endo is the inside, and then if you're dipoblastic, if you're triploblastic or triploblastic, then you have a mesoderm between the ecto and the endo. Now, a body cavity is a colon. Think of a colon like if we slice you open and look inside, what are we going to see? If we see the body cavity to exist within the mesoderm only. So let's take a look at this, because it's going to be a little confusing at first. So ectoderm is on the outside, right? That's easy. Mesoderm, space, mesoderm, right? And then endoderm. So the space is surrounded by meso. That is a collimate. If the space is between the meso and the endo, That's a pseudocolumate. See, the previous one had the space in the meso. This is the space between meso and endo. That's a pseudocolumate. An acolumate, there is no space, right? A being opposite. So columate, the space is in the meso only. Pseudocolumate, the space is between the meso and the endo. And then acolumate, no space. You guys all know, I'm assuming if you have a question, you can unmute yourselves and ask me, because I really have no way of knowing if you have a question unless you ask the question. And I would say there are no stupid questions, but there's definitely no question you should not ask. That's for sure. So body cavities have tons of functions. They are places to put fluid. So because if there's fluid inside, it'll stop the organs from moving around too much if there's impact. So it acts almost like an internal skeleton in a way. The cavity will also enable spaces to grow and swell. So like our brains, when we have brain swelling, it's a big problem because there's no space to swell. There is no column in our brain, right? It's an acolumate, I guess, part of your brain. And since there's not a lot of space for swelling, what ends up happening is once you start to swell, it goes badly quickly. So these are body plans to help be able to classify an organism. If you find an acolumate that's bilateral symmetry, then you have an idea of what you're looking at, right? And so like if there's a question, you find an organism that is acolumate and whatever, then that'll be important and you can then, begin to answer that question. Okay, so check it out. This is where that blastopore thing comes into play. So the protostome development versus the rootostome development. These are two different types of animals, right? So these are mollusks and annelids. These are chordates and echinoderms. We are chordates. I just lost it. It's back. So we are chordates. And let's look at this here. So for the cleavage, for the 8-cell cleavage, you see how if I follow this line, I'm interrupted. I have to come down this line. That is spiral and determinate. Whereas here, it's radial and indeterminate because I can kind of follow the line straight through and I don't have to turn at all. Okay. It's not better or worse. It's just different. The column. The columns here, right, are found in the mesoderm. The column here is found in the back of the actual chamber that's being created, right? So the location, this is called the archenteron, the actual spacing in here, right? So this is found inside of the blue, right? Surrounded by mesoderm. This is against the back wall where the column is being made. Okay, and there's your blastopore, the opening. For other organisms, the blastopore becomes a mouth. For us, the blastopore becomes an anus. So the fate of the blastopore for us is the anus, right? So again, neither one's better or worse. It's just different. Questions tend to come up out of this. And with this table, you really should have no problem fighting your way through it. So here you have, again, and they do this at the 8-stage cell where the lines here are not consistent. You kind of have to like bounce here and then go here. Same here, right? If I'm following this line, I can't just go through here. I'd have to come back through there. Whereas on the doritostome, you can go and follow one line all the way up. And you could also follow one line across, but so the archenteron, here's where your column begins to develop in doritostomes, in the back of the archenteron. Here on the protostome, it develops within the ectoderm, right? The mesoderm kind of develops within the ectoderm. For us, it develops more within the endoderm. And then what happens to the blastopore, right? Oh, does it become an anus? Does it become a mouth? And those are the three major things that we look at when we're trying to classify organisms. And what really sucks about this is you can't do this if the organism's alive and like walking around in front of you or swimming around or whatever. You're talking about a process that is microscopic within the development process of the organism. If you can't get a microscope in there to figure it out, you cannot know. That's a pain sometimes. So phylogenies are now based on whole genome analysis, morphological traits, ribosomal RNA genes, Hox genes, protein nuclear genes, and mitochondrial genes. Once people who have been like classifying organisms based on whether their bud is a whole or whatever, once they die, I don't think we'll go back to this again. I think we'll move on and not use that anymore. But for now, it's like a combo deal, you know? So all animals share a common ancestor, protist. So, but that's true for everything. Before the first one ever came around, there were none. There was something, but there were none of those. And then those things came around. Sponges are a sister group of other animals. They're like that in-betweener. Eumetazoans are like sponges are not eumetazoans, but animals are because they have true tissue types. Most animals are bilateral. You can be either an invertebrate, which means you have a backbone, or a vertebrate, which means you do not have a backbone. To say that again. So invertebrates, no backbone, right? Vertebrates, backbone. Now, I want you to understand that just because if you're an invertebrate and you don't have a backbone, doesn't mean you don't have a spinal cord, okay? That is a little misleading with chordata, because chordata, you think, oh, that must be a spinal cord. There are organisms that have what is similar to a spinal cord, a bundle of nerves traveling down their dorsal side, but are not organisms that have a backbone, so they're invertebrates. You see this word here? Lophotrochozoa. See, there used to be a time where students in my class used to have to know that Lophotrochozoa and what it was. Wait, does that still exist here? Yeah, it still does. All right, we'll get to it. It's fun. It's fun. It's not that bad though. But Lophotrochozoa is an awesome word to try to say 5 times really fast, because if you do say it 5 times really fast, then Beetlejuice comes or something. So these are the major groupings, phylogenetic tree groupings with some timelines in there showing you how organisms came about, their relative relationship to each other, and how these organisms, came to be, if you will, and then the groupings that they're in. Like Doretistomes are called spiny skins. The Lophotrochozoa and ectozoans, which we'll talk about each of these in a bit. So Doretistomes, well, the bilaterals are separated into 3 clades: Doretistomes, ectozoans, and Lophotrochozoans. So these clade includes both vertebrates and non-vertebrates or invertebrates. Well, and that should have been given away because it has chordates in it, right? And if you ever had to find out are echinoderms a member of Doretistomes, you would just look that up at this point. So ectozoans and Lophotrochozoans are composed entirely of invertebrates, and what they do is they have an external shell that they shed, right? They kind of lose their external shell as they grow. Because if you have an exoskeleton, right, as you grow, your exoskeleton quickly becomes too small to fit your body. So you need to shed your exoskeleton so that you can continue to grow. And then, so that process is called ecdysis. And that's how you shed your old skin and make a new one. Lophotrochozoans have to have one of two things. They have to either have a lophophore or have a trochophore larvae. So the lophophore is a feeding structure. It looks a lot like this. And they kind of open up and close and open up and close, and they kind of like capture whatever floats by. And the larval stage here, the trochophore larval stage, looks a lot like this. So if you can do lophophore feeding or trochophore larvae, then you're a member of lophotrochozoans. If you are able to be a lophophore and a trochophore, then you're a special kind of lophotrochozoans, where you can have both the lophophore and the trochophore at the same time, being a lophotrochozoan. So this lophophore and then the trochophore is actually a mophophore. All right, we're going to cut this early because I've given you a lot to digest. Let me see where I am with this. I'm gonna stop the recording here. Well, let me stop the share.